Hello, everyone. Thank you for you being here today. So this morning session is about advanced clinics and the use of all types of electrophysiological signals in clinical situations, ranging from epilepsy to autism and consciousness disorders. So the first speaker is Jean-Philippe Lachaux. He's the INSERM research director and the co-head of the EduWell team at CRNL. And he has more than 20 years of experience dealing with oscillatory brain dynamics. He's a gamma dealer, and he will talk today based on intracranial recordings. And he will talk today about what he has developed during the Human Brain Project, and which continues today again. Okay, Jean-Philippe.
Thanks, Olivier. Merci, salut à tous. There we go. So hello everyone here and abroad. I will tell you about software. It's called EBOP, which is designed to visualize intracranial EEG data, and which was a deliverable of the Human Brain Project. So it's been developed over 9 years, 10 years. But also I will take this occasion to explain you the motivation for this software first, which I think is important. And just to start, because maybe some of you are not familiar with intracranial EEG. So the starting point of intracranial EEG is usually a patient, epileptic patient, doing an experiment, as you can see here. So pushing buttons when seeing faces or landscapes or whatever. And then you get recordings from parts of her brain, so the little bubbles that you see there. So the spatial resolution of those recordings is on the order of a few cubic millimeters, and then you get a measure every millisecond. So you get high spatial and temporal resolution, which is one of the main reasons to do intracranial EEG recordings. And usually the characteristic of those recordings is that you end up with very strong observations. So that would be an example where, for instance, on the left, what you see is the what's called the gamma band response, so increase of energy in the broad frequency band between 50 and 150 Hertz, just an example, to show you how the energy increases when you show pictures. So the horizontal, vertical lines corresponds to different pictures of different categories. And this is time horizontally, and you can see, for instance, in that case, in the fusiform gyrus, very strong increase to faces. So what I mean by my point here is that as Isaac Fried was saying once, you don't need to hire a statistician for 1 month to just make sure that there is a response there. Usually it's pretty obvious. And in most cases, we are interested in very major effects. So that would be a characteristic of this kind of approach, and I would say it's very much like diving. So I would call it the deep dive approach, meaning that you're concentrated on one patient, one particular patient, and you will find something very relevant and very obvious at one point. So it's like diving and finding the Titanic, for instance. It's an approach that is really based on one individual, and that is very common for clinicians. Because if you're a clinician, you want to understand one patient. And also it's very common to in non-human electrophysiology, as you know. Usually papers are about 2 or 3 monkeys or cats or whatever. So you find you're already looking for very obvious effects. For me, it's very much like sending a probe to Mars, like Curiosity, and then let's say that you get this little rover and it finds water. So you go to that planet and then you find water. That's an observation. It's a very strong observation. It's 1 or 0 thing. And then after you, well, it's basically about exploration. The question that you get then is obviously the problem of generalization. Because once you made a very nice observation in one individual, how do you translate to the global population? And it's tricky, and it's not even sure we're completely clear about the meaning of that question. What do we mean by generalization? Do you expect this result that you found in Mr. X or Mrs. Y to be true in every single individual, every single human being? Usually that's obviously unfeasible. So what exactly do you expect? But again, it's a question that is not unlike what you find in non-human electrophysiology when you record, let's say, from, I don't know, the lateral, the intraparietal sulcus in a monkey, or in 2 or 3 monkeys, and you find something. And what can you do with that? If you read a paper like that, what can you do? Does that mean that if you record from any single monkey in the world, you will find that effect? Obviously not. So it's kind of a gray area where you don't want just one monkey, or you just don't want just one patient. You want more, but you don't know exactly how much you should replicate that effect. And actually, that question has been addressed by 2 papers. One chapter that I wrote, and then I found that paper from Pascal Fries and Eric Maris. What do you do when you have very small samples? So here they talk mostly about monkeys, but that can be patients. And it's not clear exactly how many patients, how many monkeys you should have. So what the essential element here or parameter is typicality. So you want your result to be typical. That means it's not an anomaly of your sample. It's something that you would find in other individuals. But typical means that you should find it in a certain proportion of the population. Certain proportion meaning that you get a percentage, that you have to define. So for instance, Pascal says that it takes 50%. He said, I want my result to be true in 50% of the population. And so it comes to that estimate of 5 individuals. But 50%, when you think about it, if you find a very, very nice mechanism and you can see that it's true in 50% of the population, I mean, what do you do with that kind of result? Because that means that maybe you have 50% of the population that have a brain that works completely another way. So I don't think this 50% is very valid. And in fact, what you should shoot for is maybe 95 or 100%. But that requires a number of individuals that is just huge. So there is no clear answer to the question of how many patients, how many monkeys should show the effect that you're interested in. I came to the conclusion that it's 3, but it's really a matter of debate. Anyway, so that's one thing. At the same time, there is a change in the field of intracranial EEG recordings, where now it's become more and more easy to get many more patients. And it's partly due to better organization across centers. Now it's not uncommon that you have a paradigm, your paradigm, that would be distributed to several centers doing intracranial EEG recordings, and then you get data, and you can easily get 50, sometimes 100 patients, but and sometimes in many tasks. So the difficulty of that is that you get a complexity of data that we are not used to. Because of course, and also you have to know that the position of the electrodes from one patient to the next is variable. So you get all those recording sites that are all over the brain, very variable. You get so many tasks, and what do you do with that? The question that, sorry, I come to that point after. The question is how you stick to your idea of deep dive, of really focusing on one patient and really looking exactly in detail of an individual and that particular effect when you get so much data. You might say, and then there is a shift really in the field, in that more and more people coming from fMRI research or EEG or MEG basically come and say, well, we know how to handle a large group of subjects because that's what we do for a living. So the shift is between that approach that was really based on very similar to non-human primate, for instance, electrophysiology and clinics, to this approach which is more at the group level and based on the methods of fMRI and EEG and MEG, which is basically forget the individual, we want the big picture. And we know how that works. I mean, if you get MEG recordings, for instance, what you can do is that you take one particular sensor, and then you can average, just keeping the same sensor across individuals, or you can do that with voxels in fMRI or with EEG electrodes. Okay, you get a way, because you have labels, to group the data, to pull them together. When you come to intracranial EEG, it's not that easy, because if you want to reproduce the same approach, basically you need to say that this electrode and this one in several patients, just the same. Okay, so we put them together. And right now the approach is using parcellization and atlases. So you would say, okay, this electrode is in that particular area, and in that other patient also, and in that other patient also, and so on. So I put them together, and then I do statistics. And then I can use the statistical methods used in when you get to deal with groups. But the problem, and that's my conviction, is that you really lose something when you do that. And that's the main motivation for the software I will show after HIBOP. Because if you do that, and again, this approach becomes more and more popular because of the difficulty to deal with this very complex and very big data set, I think you lose something. Let me show you that example where you see this is the same patient in one task that I don't want to detail. And you get a response. Okay, well, I don't want to detail how it's shown, but get this response. And you move by 3.5 millimeters to the other red spot. And the response is completely different. That's intracranial EEG. Is that you move by 3 millimeters and you get a completely different response. So if you do that, I don't want to pull those two sites. I don't want to put them in the same global. I don't want to give them the same label. And I don't want to do statistics, pulling them together, because for me, it doesn't make sense. So that means at some point, you need to have really access to the individual anatomy. So how do you reconcile the deep dive and the big picture? What do you do with that? For me, but again, maybe that's personal, I'm convinced when I see an intracranial EEG study, when the paper tells me, basically, if you record here, you will see this. Okay, go there, take one patient, just do this task, record, and you will get this. That's what you will get. That's what I want to see. That's what I want to get from an intracranial EEG papers, plus variations about connectivity and whatever. And in fact, when you think about it, it's like a treasure map. Because a treasure map tells you, if you go to that island, if you go past the trees, past the little dune and so on, you dig, you will find that. You will find the treasure. So that's exactly a treasure map. So in my opinion, most intracranial EEG papers should be treasure maps. That's what I like in an intracranial EEG paper. So what would be a treasure map? In fact, it would be something like that. If you record, let's say during the rehearsal of a verbal working memory task, so very specific about the condition, a site that lies really in the depth of the inferior frontal sulcus, for instance, really in the depth, not superficial, right there. Okay, and you know that this site is also deactivated during visual search, so you had a constraint about the function in other tasks, like using localizers. And you know that this site is also anatomically connected to POPO, because you got this information from, let's say, cortical evoked potentials. You get that. And so on, and so on, and so on. Then you should see this. That means that you try to be as precise as you can in the definition of the region that you should record from to find this effect. And then you start understanding something. If you don't get that information, if you just say, okay, you're in the inferior frontal gyrus, boom, okay, then of course you will get a huge variability. Some sites will show the response, some others. But if you're more and more details, then you expect to get something that's more and more robust. So that's the idea of a treasure map. Okay. And of course, then you can deal with this information getting from many patients, because if you get just one patient, okay, you get some information. Okay, I get the effect here, but not here, not here. But as you get more patients, of course, then the maps become more detailed because you get more information from other patients. So you do statistics, but in fact, the power of the group, you use it like that to, in fact, get maps that is more and more and more detailed. But there is one very good reason not to do this, and why nobody does that with a large group of patients, because it takes years to process that, because it needs very, very detailed investigation at the anatomical level of every patient, and you need to repeat that to all the patients, and so it really takes years. So it's unfeasible unless, and that was the purpose of HIBOP, you create a software that is specifically designed to do that. So fortunately, we had the money for 10 years, really, to invest seriously into a professional level software. which is based on Unity, for instance. Unity is a standard in video game industry, and this is to facilitate the interfacing and the interaction with the display. The brain is very fluid, and and now I will show you how it works. So this is just one way to use eBop. There are others, but I think it's very illustrative. And basically, okay, let's say you recorded from 57 brains during an attention task. Okay. So what you can display whatever you want, raw data. What you will see here is band-limited power in the gamma range, but it can be also the result of statistical analysis, whatever you want. And then, okay, very important is that you get very, very easy and fluid access to the dynamics of the task. Okay. You don't want to go and to have to go through, I don't know, before that, I had the same problem and my solution was to use MATLAB and to create movies. So, okay, I would run my script for half an hour, create a movie and then watch the movie in quick time. Okay, boom. No, you want something that's really much, much more fluid. Okay, you want to get your instant visualization of the global dynamics over all your brains. And then obviously, so you see this increase in red of activity in certain regions of interest that you will define. And okay, so you define a region of interest. You say, okay, there is something that is massive here, so I want to investigate this region in more detail. Then what you will do, put your little bubble, whatever. Again, this needs to be convenient. Sometimes you need to go in the depth, so you need to remove part of the cortex if you want, let's say, to access the insula. The insula is hidden, it's behind, so you need a way to erase part of the mesh to access that and put your region of interest. And then you want to visualize all the responses that you get in that region of interest. So you get your sites. And then the process will be mostly an investigation of all the sites one by one and to understand the variability, basically. So you see, okay, well, in this case, it's mostly a deactivation. You get a suppression of activity, of gamma activity, okay? And then you will see some signs that are completely atypical. Some are very typical from the group, and some are very atypical. And then you will use the ability to go very fast to the individual level anatomy to find reasons for that deviation from the norm of that region of interest. So this is really, you take your site, you just click, and then you get the individual patient that comes, and you can see exactly where the site was in that patient relative to that particular sulcus. Okay, is it in the depth, more in the superficie? Where is it exactly? And then basically you learn your anatomy doing that. And then with that question, why does that one shows an effect? Why not that one? So one thing that you can get also is you want to get instant access to additional information. So you want something as easy as just moving your mouse around and on your site, and then you get all sort of information. So here it's minimal, but you can get also the effect of stimulation. Okay, when that site was stimulated electrically at 50 Hertz, we got that effect in the patient. The patient stopped talking, for instance. Or you can get information about the epileptogenicity of the site, like there were many epileptiform activity, so this one is more like probably contaminated by pathological activity and so on. So you can have all the access to many information that you have here. And then the idea is to create groups of IEG sites that are homogeneous according to a certain number of parameters that corresponds to the treasure map. So you create your group, and eBop is not designed to perform analysis. It's just designed to create those groups and export them. So that you would do simply once you have your labels. So you labeled all the sites of interest, you give them a name, you export, and then as simple as that, you get a tab delimited file with all the information you need to run any script that you want. In the script, it specifies where the data are, coordinates, and so on. Boom. That's you do your statistics at that point. Just to finish, just to show you practically, I know it's very superficial in terms of presentation, explanation, but to give you the big idea, how to get into eBop. Well, if you want to analyze intracranial EEG data, what you need is a correspondence between the traces that you recorded during your task and the location of the electrodes. That's what you need. you need to know the correspondence between the anatomy and the functional data files. So you provide them, you make the correspondence, you say, okay, this is where I have my anatomical files, that's my MRI, that's my mesh, and so on and so on and so forth. And then you say, okay, my data are here. Boom. Very easy. It's in the bits format so that it's maximally compatible with any kind of data storage that you have. Also, another thing that you need, I mean, as you know, if you do experiments, when you have traces, usually you send events, pictures, whatever. So you need a file that describes exactly what the events are with their meaning and how you should cut and epoch your data. So this is specified also. So there is a specific format for that, but there is an interface to create that, to create your protocol file. That's again pretty obvious. And then you specify what you want to visualize. So in that case, I showed that particular example because to show you that you can visualize side by side. So either intracranial EEG data from several experiments, because sometimes it's very useful to get to compare in the same patients data from several experiments. And also you can display MEG data, EEG data, fMRI, cortical evoke potentials, just anatomic, and of course intracranial EEG. So again, you can have that side by side. So it's very multimodal. What else? And then also you can tell HIBOP to find where to find additional resources. So for instance, we had a long-standing collaboration with Bertrand Thirion at INRIA, who's done something pretty incredible, which was to record the same subjects, normal healthy subjects, I think 10 or 12 of them, in a number of tasks that is just maybe 60, I don't know, it's just enormous. So all possible tasks in the same subjects. It's called the individual brain charting project. And the end of that, the end result of that is a series of contrasts that shows you exactly. So for instance, here you see the effect, the contrast between saccades, making saccades versus fixation. And so you can display the sort of map on top of your electrodes. Of course, you can also display atlases if you want. So you can do that. Okay, and just to end up, because IEEG exploration, so I told you at the beginning, it's complicated, it's complex, many data, and so you need to make it fun. And for me, it's not a gadget to be able to use virtual reality with the HoloLens system to really manipulate your brains in 3D. And this is an example of what we did. So basically, you get your brains, and it's like, so you get your environment of work, your workspace, and basically your brains, they float in space, and you can make them as big as you want. So I was very interested in the orbitofrontal cortex at some point, and I worked on the location of the precise sites on a brain that I made maybe 5 meters wide. And I tell you, it's your relationship to anatomy is completely different, because already it becomes a planet. I mean, my memory of the anatomy and my interaction and my relationship to this particular brain region has completely changed by being able to manipulate in 3D in space, just have my brain here and walk around basically and see, okay, I have an electrode here and here and here, and then display the activity. So it's not a gadget, it's really something that is very important. And we can do that with setups like HoloLens or whatever. So we can do that too. And let me just thank you. So one very important thing, so you can get it here, so it's all free. And also now HIBOP is intended and already accessible to the HIP, which is the Human Intracranial Platform, which is a deliverable of the Human Brain Project, which is a project to collect data across centers, intracranial EEG centers, just mutualized tools that can be used by anybody. So it's in the HIP too. And of course, so I thank the developers of HIBOP because I couldn't write a single line of C#. Thank you.
Okay, we have time for questions. Yes, Christian.
Yeah, thank you very much. So what about clustering the responses across patients to handle this variability? Because as you said, it can be variable by a few millimeters. So trying to find reproducible patterns across patients.
Yeah, well, that's the idea. How do you cluster? And what I see mostly is using anatomical labels, which means that you rely on an automatic parcellization, for instance, by FreeSurfer or MassAtlas. And can you really completely trust this automatic labelization? I would say that maybe for most of the sites, but sometimes not. So I really like to have that immediate access to the individual anatomy just to check. And also, this example I showed was not on, was really on purpose. This, when you really deviate by 3 millimeters, because you can see, I'm pretty sure that pretty much every anatomical labelization would have put it in the same parcel.
On the temporal time, on the time course, for example. Clustering not anatomically, but functionally.
Oh, okay.
On the gamma band response or the set of gamma band and evoke potential.
So in that case, you would look for you pull together sites that have the same time course?
Yes, okay. For example.
But then what do you do? Because often, as you know, you find responses that have the same time course, one in the fusiform gyrus, another one in the FEF, for instance, or whatever. So you need to, anatomy has to come into play at some point also.
Okay, thanks.
Other questions? Oh.
So it's great because it's a relief. My engineers are not here, and I feared any technical question. Thank you.
Okay, thank you. So we continue with Pauline Mouchès, who is a postdoc with Romain and Quentin in the dual team at CRNL. So she will talk about epilepsy again and deep learning methods for epileptic spike detection in MEG. Pauline?
Hello, everyone. So today I will talk about automated spike detection in MEG recordings of epilepsy patients. And so this work is done in collaboration with my colleagues Thibaut Dejean, who's doing an internship with us, Julien Young, Romain Bouet, Carole Lartisien, and Romain Quentin. So let's go through a first quick introduction of MEG in epilepsy. So the motivation for this project is that 30% of epilepsy patients are drug resistant, and in these cases, surgical treatment is indicated. And the success of the treatment relies on the accurate localization of the epileptogenic zone from which the seizure arises. So this pre-surgical evaluation is done using many different imaging modalities, which can be invasive or non-invasive. And especially magnetoencephalography, which is a non-invasive method, has been shown as being very efficient to help localizing this zone from which the seizure arises. So magnetoencephalography non-invasively records neural activity with a high temporal and spatial resolution. And in MEG recordings of epilepsy patients, we can see interictal spikes. So you have an example here in the red box that actually originate from the epileptogenic zone. And once we identify these spikes in the recordings, we can use source localization, source reconstruction methods to localize the source in the brain anatomy. The main problem is that detecting these spikes is usually done manually, and it's a time-consuming and burdensome task that is usually done by the clinicians. So for this project, we're interested in this first step of spike detection, trying to automate it. So there are a few already existing methods for this purpose. And I would say the first two kinds of methods, which are called processing-based method and traditional machine learning methods, they are based on characteristic typical characteristics of MEG spike morphology. So you can see here on the figure, for example, we have a template of a spike. So spikes have given amplitude, duration, etc. And so the first type of method, which is called processing-based method, are, for example, amplitude thresholding or template matching based on the shape, typical shape of spikes. Then traditional machine learning methods were proposed, and they usually follow a 3-steps process. First, preprocessing of the signal, then feature extraction, and finally classification. And so features are that are usually extracted from the signal include amplitude, curvature, slope, duration, etc. And then once we have all these features, we feed them to classification models such as logistic regression to define if the signal contains a spike or no. The main issue with these 2 types of method is that they rely on a priori knowledge of the shape and characteristic of a spike. And so the human need to design these features prior to running the method. However, one additional problem is that spikes are pretty variable across participants, across patients. So you can see here examples of 2 spikes of 2 patients. They don't look exactly the same, so using a priori features is not optimal. And that's why recently a few methods were proposed using deep learning. So deep learning allows to automatically extract optimal features for the task. So we don't have to give any a priori information. The model does learn the information by itself during the training. So there was a few methods proposed. However, they mostly rely on complex annotations, so channel-wise annotation of the spikes. And they were mostly tested on simplified data, so only typical spikes were included. So to overcome this problem, we proposed a method based on deep learning. And we have a few requirements for our method is that we want to have something that is fully automated so that it allows generalizability across patients, across machines, etc. We wanted to work on simple annotations so that we can have continuous model improvement. So if clinicians can simply annotate new data, then we can use it to fine-tune our model. And the last one is more of a clinical requirement, is that clinicians usually look for methods that have a high specificity, which means that they do not detect too many false positive spikes. So let's look at the data now. So we have data from 95 patients. Each of them have 45 minutes recordings, and we have a bit more than 4,000 annotated spikes. So you can see here on the figure example of recordings with a few spikes annotated. As for the preprocessing, we did a very simple preprocessing, just bandpass filter resampling. And then we created small windows within the signal. So as you can see in the red rectangle here, we cropped 200 milliseconds windows within the signal, and each window was labeled as containing a spike or no. And so then there's a few important considerations to account for before designing our model. The first thing is that spikes are focal events, so they appear on adjacent channels, but you don't see them on the full channels. So we can see an example here where the first 2 spikes are visible on the top channels here, but not on the bottom one. Channels are just ordered by region. And this is something that past deep learning methods did not really account for. So the question is, can we account for the 3-dimensional spatial relationship between the channels, between the MEC channels in the model. So basically the three-dimensional spatial localization of the sensors within the MEG helmet. Second consideration is that you can imagine that when we crop these small windows, we end up with way more windows that are spike-free compared to windows that contain spike. Actually, we have one window displaying a spike out of 80 windows, which creates a very imbalanced data set. And this is problematic in machine learning in general, because it's pretty hard to train model when we have imbalanced data like this. So to start on a simpler basis, we simplified the problem and subsampled non-spike windows to have a balanced data set. So that's just to start with. Basically, now I'll describe how we designed our approach. So based on this first consideration, we decided to represent the data as a graph, as you can see here, and to use a graph neural network to train a model that can classify graphs. So basically, one graph will represent one 200 milliseconds window, and then we can classify graphs as containing a spike or no. So for graph neural networks, the graph is defined as this and has three main components. So I don't know if you can see my window here. Yes. So the graphs have nodes, which can simply be defined as the MEG sensors. Then the graph has features. So basically, each node in the graph has a feature vector that contain information that represent that node. And finally, the graph have edges that can be weighted and that connects nodes. So as for the nodes, pretty straightforward. We just use one sensor for one node. As for the features, we first thought that we could maybe just use the raw signal time point value for the feature of each node. So if we go back to that one, for example, for the top channel here, we could just use this part of the signal as a feature for the node. And then we thought maybe a better way to design our features would be to actually stack another small deep learning model here to create a compressed representation of the raw signal. So the raw signal contains artifacts, is noisy, etc. And by using this first block in the model that takes as input the raw signal and outputs a compressed representation of the raw signal, we already extract important information regarding the spike classification. Then for edges, so to define the edge weights, we thought about first simply using the distance between MEG sensors within the helmet. So it's not exactly the distance, but the inverse of the distance. So sensors that are close together in the helmet will have high weight connection, and sensors that are far away in the helmet will have lower weights on their edges. That's what we can see here. And blue sensors that are in the same cortical area have higher weights. Or maybe another way to define these weights could be to use the connectivity between sensors. So we could compute the correlation between the signal between the signal of each sensor and use that as edge weights. So after a few trials, we figured out that the best parameters for our models were the following. So we have 274 nodes, which are the sensors. Our features are the time points that are pre-processed by this first. So this is a convolutional neural network, but can just call it a first deep learning model block. And then our edge weights are the distances between the, well, a function of the distances between the sensors. So this is just another way to look at the model from start to the end. So we have the raw MEG data as input, then we have our preprocessing. This time CNN is just this black CNN block. Then after this, we just reshape the data into the graph. We have a few graph convolutional layers. And then this graph model allows us to do the binary prediction. Does this graph contains a spike or no? And then we can go back to the full length recording of the patient and display the output for each small 200 millisecond window. So let's now look at some of the results of this model. Okay, so here, so we compared our model to the two state-of-the-art deep learning methods. And these are the first two rows of the table here. Third row of the table is basically this CNN block without the graph part. So we are not using the graph part, we're just using basically the features extracted from the CNN to make the decision. And then the last row is the CNN with the graph. So we can really see if adding the graph and taking into account the spatial organization of the sensors is actually helping or no for the classification. So here we trained our model on balanced data, as I said, and here we also test on balanced data. So it's not realistic data. And the observations we can make are that using the graph improves the scores. So we have the accuracy F1 score, which is basically a good representation of specificity and sensitivity together. So the higher, the better. And then we can observe something which is pretty interesting, is that sensitivity is higher when we use the graph, which could be interpreted as the fact that graph edges bring complementary information that allow to detect more spikes that maybe have lower amplitude or are less visible in the signal. But we also see that the graph model has lower specificity, which means that it's going to detect more false positives. And then we move to realistic imbalanced data. So basically, we kept our model trained on our balanced data, and then we used a little trick, I would say, to adapt it to imbalanced data. So basically, the main problem is that if you train your model on data that have a specific distribution and you test it on test data that have a different distribution, you'll have a mismatch which cause the model to not perform very well. So we just use the threshold moving approach to compute these results. And the main observations here is that we have a big drop in performance. We can just look at the F1 score here, which was around 75 before, dropped to 25, but that is common as we have more windows, more artifacts, and also the model was not really optimized to work on this imbalanced data, but that was just a first test. And what is interesting here is that we still see this specificity, sensitivity difference between the CNN only and the graph model. So we still have our graph model that is able to detect more spikes, but it still has lower specificity, so it's still detecting too many false positives. So as a conclusion, we saw that deep learning is a promising method for interictal spike detection. The graph allows better or more natural representation of the data and gave promising performances. And the developed methods can be applied to other data or use cases we could imagine that we can use that for any event detection in MEG or EEG signal, for example. And as future work, we would like to include more patients to increase the data diversity so that the model can learn from more diverse spikes. We would like to optimize our methods for working with imbalanced data. So trying to develop methods for training the model on imbalanced data directly. And a last idea would be to move to an object detection approach that would avoid having to create these windows within the signal, and maybe just looking at the full recording and being able to automatically detect the spikes within the recording without having to crop the windows. That's all I have for today. Thibault Lejean, who's doing an internship with us, made a poster. So if you have a more detailed question, you can go see him at the poster break. And thank you for your attention.
Thank you, Pauline. So any questions?
Thank you. Hello. I would like to know maybe if you use a pre-trained backbone, or were you able to train your model from scratch?
Did I train the model from scratch?
Yeah.
Yeah, all the models are trained from scratch. The two models that I listed as state-of-the-art, I also just took the model architecture, but trained them from scratch on our data.
That's the form of the spike depends on the type of epilepsy, and you could separate the groups.
I don't think that I'm not an expert in epilepsy and spikes and all that. I don't think the shape of the spike really depends on the type of epilepsy. I think it's more the localization, so on which sensor you're going to see it, if you have temporal epilepsy or, yeah.
Thanks a lot. So did you have a look back at the false detections to try to get a feeling? Because you said it's mostly artifacts. It's like spiky artifacts.
Yeah, we're currently trying to do that. Trying to do also some spike clustering to see to understand what the models looks at to make the predictions, basically. Yeah, that's what we are currently doing.
So you showed that you use the inverse distance instead of using the connectivity, and that having a graph model may bring complementary information compared to the other model. Do you think that using also the connectivity instead of the inverse distance could bring also another kind of complementary information? And have you tested that also?
Yeah, I think it could help. So far, we've tested only just using the correlation between the signals. It didn't help much versus the distance, but I think there are more complex connectivity measures that we could investigate, and we should definitely do it. So yeah, that's also something that we want to try to do.
Thank you.
Other questions? So maybe I have a question for you. So you wanted to integrate the several channels, adjacent channels in your analysis. What about going to the source space?
Yeah, that's also a very good point. We didn't. So that was the next step of the project. First, there was like automated spike detection, and then going to source and maybe see if the sources we detect with this automated method matches with the sources that were detected with the clinicians. So that's also something that we need to figure out. But yeah, that's.
What are your expectations? You think this will improve?
I think it
Or
could
you have-
improve. I think it could improve. In general, the more information you give to the model, or the more good information you give to the model, the better results you obtain. So, yeah.
Okay.
Yeah, I had one question regarding the fact that you still need to label and epoch the training data as spikes before using the deep neural network.
Mm-hmm.
And have you thought of doing like unsupervised machine learning, such as HMM, for example? Maybe it would also work.
Yeah, that's a very good question. That's true that we have some patient data that are annotated. We also have some new patient data that are not annotated. And we thought maybe not going completely unsupervised, but maybe doing some semi-supervised training. So having one part of the data that is annotated and another part that is not annotated, but still contributes to the model training. So that's something we thought about. I also thought about going completely unsupervised and doing some kind of anomaly detection and expecting the spikes to be detected as the anomalies. But the fact is that there are a lot of artifacts in the signal, and I think that going completely unsupervised, it would catch all the artifacts. So yeah, I'm not sure it's a very good idea to ignore all annotations, but definitely semi-supervised could be interesting. Yeah.
Okay. Thank you.
Thank you.
Other questions? No. Okay, so thank you, Pauline, again. Then now we move to Françoise Le Kenya, who is in some research engineer working on the coffee team at the CRNL, and she has developed advanced analysis and models for analyzing MEG recordings in autism.
Thank you, everyone. So yes, I will talk in this presentation. I will talk about Bayesian approaches for perception and how they could help to address autism. So I just want to say that I'm clearly not an expert in autism, but hopefully, fortunately, I have colleagues and friends in the audience, Jérémy Matou and Lauriane Sapé-Triomphe, and there's also Marie Gomo. She's not involved in our work, but definitely an expert in autism. So I'm glad they can help if you have a tricky question. Okay, so I will start with this kind of provocation. Shall we forget about these approaches for autism? And I'm asking this question because this year there's a very remarkable review that was proposed by Angeletos Chrysatis and Series, and they did a great job to review 10 years of Bayesian theories of autism. And this is what they write. The results of the studies have been mixed and occasionally conflicting. So that maybe we could stop this presentation and go to the coffee break. But I propose that we dive into this Bayesian approach for autism so that we can discuss this. And I'll go back to this review at the end of this talk. And definitely what they claim is not to forget about it. So this is the outline of this talk. First, autism, then then a Bayesian approach to perception in neurotypical participants, then wrapping up together, and then we'll see. So first, autism. So actually, we should speak about ASD, autism spectrum disorder, because autism is globally defined as a continuum. And people with ASD could represent 1% of the population. And the two main symptoms that you may have heard about pertain to some difficulties to understand social interactions, like difficulties to understand emotion, interpreting a gaze or a smile. And also, they have interest in restricted or repeated behaviors, like being passionate by trends, for instance. But these are two, I would say, big pictures. And as Series and her students describe in their review, it's a much more complicated picture. For instance, regarding sensory processing, there are much thinner symptoms associated to ASD, like hyper and hyposensitivity, increased attention to details, and others, as you can see. So in the end, it appears such a challenge to build up a theoretical framework that could account for all these symptoms altogether. And yet Bayesian theories of brain function, this is what they attempt to do. And in this talk, we will see what it is about for perception, where perception is seen as Bayesian inference and learning. So first, Bayesian learning. So we will consider Bayesian agent that can be the brain, as we will hypothesize here, but it can also be a robot or any kind of system processing information. And the idea is that this Bayesian agent collects some observation, and the aim is to infer about the hidden causes of this input. So for instance, if we consider this knot, the hidden cause of this knot is the musical stream, the melody of which this knot belongs to, and so the hidden rule that the system would infer is the specific transition between knots. And to infer about this hidden cause, the system would be equipped to an internal belief of how the world, the world generates this inputs, and these are called the prior beliefs, and they are built up over past experience. So prior having collected this information, the system is in this state of knowledge, then collect the information, and by applying the rule of bias, computes the updated version of this knowledge, and this is called the posterior belief. And this is a learning framework because posteriors here, the updated, the revised internal knowledge, become the priors for the subsequent future data collection. So we work in a probabilistic framework, meaning that information are represented by a probability distribution. So contrary to logical theory, an information is not either true or false, either 0 or 1, but somewhere in the middle. And that means that in the middle, it's about plausibility, how plausible, how reliable is the information that is manipulated by the system. And this enabled the system to process differently reliable information contrary to unreliable ones. And this has to do with an uncertainty account. So for instance, if we consider a system that should process this stream of this musical stream here, then as you can see, the same sound sequence here delivered in two contexts, this noisy one and this quiet one, would not be attributed the same precision of information. So applied to ASD, it's about the hypothesis is about an impaired Bayesian inference. So the first proposition was proposed by Pellicano and Burr 10 years ago, and they proposed that the major symptoms of ASD could be explained by hypopriors, meaning that whenever the people with ASD would collect information, they would be more influenced, they would give more weight in this balance with the prior and the sensation. They would attribute, no, it's, sorry about that. They would assign, they would give a lower weight to the priors, so that all the incoming information would be dominating in this balance, and this would lead to a reduced influence of this prior knowledge, so that in the end every new sensation would be surprising, and this could account what is called sensory overload or sensory fatigue. And they have, following this work, there has been different proposals, and the dominant hypothesis now is called the high PA hypothesis, which means high inflexible precision of prediction errors in autism. And basically, this hypothesis rests on a more sophisticated view, where this is not either this part or this part, but it's a mix of the two, and basically it's the ratio of the two that would be impaired. And the important thing is that these mechanistic frameworks provides new untestable hypotheses that, and we will see how the scientific managed to test them during perception. So now let's move to the second part, Bayesian approaches to perception. So perception as Bayesian inference is not a new hypothesis. It was already proposed by Helmholtz at the end of the 19th century, where he proposed that the aim of perception is to infer about the unknown causes of our sensation. He talked about unconscious inference. And a few years ago, Diane and Princeton, among others, they could propose a mathematical reformulation of this psychological view. And one possible algorithm that could implement Bayesian inference has been proposed. It's called predictive coding. And predictive coding is a general learning framework based on error minimization that is of particular relevance to host Bayesian inference processing of information. And I will see in the next slide is based on a hierarchical structure with ascending prediction errors and decreasing prediction. And importantly, its biological plausibility was assessed and described by Friston and Bastos, leveraging on animal empirical evidence. So this makes this framework really relevant to test as a perceptual model. So since we are in cutting edge, I think we can take time to demystify how it works and how we can how the learning operates, stimulation after stimulation. So say consider this an agent being equipped by such a framework. And so let's say that the system has been exposed to a series of repeating observation like X. And so we're almost to be at trial K, and so at trial K minus 1, this is the state the system is. So because it's been exposed to X, the system predicts that future sensation will be an X. And then at trial K, the system collects an X as predicted. So no mismatch between the prediction and the observation. So what we call the prediction errors is equal to 0. So the update of the prediction is such that the system will also predict an X for the subsequent observation, and the precision of this prediction becomes stronger because the model is really confident that the next trial will be an X. So at trial K plus 1, when the system collects a Y, this is really unexpected. So there is a mismatch here. There's a mismatch between the prediction, an X, and the observation, a Y, and this makes a prediction error. And then the prediction is slightly updated. Like the system is not already predicting a Y because it has just upon once. Of course, this is a very schematic approach just to see how it works. And so the system will still be predicting an X, but this time the precision might decrease a little bit. And at trial, at the next trial, if the system still receives a Y, then progressively the prediction will evolve towards a Y, and if it's an X, then the prediction will still be an X, but the precision will get stronger again. Okay. So since it's implemented in a probabilistic framework, we're not working with a prediction error per se, but with precision weighted prediction errors, meaning that this prediction error term here, they are weighted by a mathematical quantity that represents the precision of these errors. So, for instance. And this gain, it enables to filter the relevant errors and to that internal belief only if the errors are meaningful in relative to the context. So, for instance, if you live close to an hospital, then the siren of the ambulance doesn't tell you much to you because you're used to hearing this kind of information. So this surprising event will be assigned by the brain with a low gain, a low precision weight, so that this error will not trigger any update in the learning, and no behavior this will not lead to a particular behavior to adapt to this situation. Whereas if now we can hear this noise, this would be very unlikely, and we would behave accordingly. Okay. So now the mismatch negativity, we will see why it is a good candidate to assess perceptual learning. So mismatch negativity is an electrophysiological component that can be measured with EEG or MEG, and it's obtained using this kind of simple paradigm. It's measured mostly in the auditory modality, and so basically, this is what we call the oddball paradigm. We have a repeating sound like this, beep, 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 beep. And from time to time, unexpectedly, we have different sounds called deviants. And when you compute the evoked response to the standard that you can see here in gray, and the evoked response to the deviant in purple, if you compute the difference between the two, deviant minus standards, here we have a big negativity at frontal sites in EEG, and this is the mismatch negativity. And this component has been described for almost 50 years now, and it's been intensively studied, described, and yet we don't know really what it means in terms of functional role and its underlying physiology. And in this context of Bayesian learning, the MMN could represent a context-sensitive prediction error. As the brain is exposed to repeating standard tones, it could predict with a strong precision standard. So when the deviant occurs, then this would elicit a precision-weighted prediction error. And importantly, this view of this computational interpretation of the mismatch negativity has led to new model-based manipulation of this component. And notably, they led to manipulate the statistical structure of the sound sequence to assess this hypothesis. The more predictable the input, I should quicker. The more predictable the input, the smaller the error. So we tested this hypothesis with Jeremy. We designed, we consider a typical oddball sequence as usual, and we contrast it with a predictable context where in this case the deviant could occur regularly. So we have two standards, one deviant, 3, 4, 5, etc. And we expected a reduced mismatch negativity in the predictable context because this would represent a reduced prediction error. And this is what we could indeed measure. We have the predictable MMN in green that is reduced as compared to the unpredictable one. But then once we have said this, we wanted to go further, and since these two mathematical quantities, they are mixed together by the multiplication into the MMN, we wanted to, as we wanted to assess their relative effect, we had to go to dynamic modeling. So in short, dynamic modeling is made of different steps that I will explain quickly. So if we consider one particular electrode, we have the first response to the first tone of the sequence here, and we have the last response here. So we have the time here corresponds to the experimental time. We have all the stimulation here. So the first step is to extract a vector time series here at a particular latency. So for instance, at 200 milliseconds. And then we consider the different hypotheses that could describe the processing of oddball sounds. And we translate each hypothesis into model, and each model will give us a prediction of single trial response, a trajectory here. And then for each model, we obtain different prediction, and we confront these their respective prediction to the real data that we have measured with our participants, and this confrontation relies on model inversion. Then we have a very powerful statistical tool. It's called Bayesian model selection to compare the different models when confronted to the data. That allows us to select the winning model, and on the winning model we can then perform an analysis of the parameter. And of course, we should consider an experimental design here, the sound sequence, that enables separating the models. So applied to this study, we extract the single time series at the cortical level. We consider a null model, non-learning and learning models with different predictions, different time series. Oops. Then we perform Bayesian model selection, and we found that every time sample, the null model was winning, but like in the green here, we see that at the latency of the MMN is depicted in red here. At the latency of the MMN, the learning model was found clearly winning, outperforming the others. And then regarding on the winning model, we wanted to have a look at the model parameters. And maybe I'll skip this part, but just we can discuss this at the coffee break if you're interested. But yeah, just one word: we are few teams adopting this approach, but we all find a robust evidence towards Bayesian learning. So now, applied in autism, I will go quick to this point, but I'm almost finished by the way. So dynamic modeling was employed in our team during the thesis of Lauriane Sapetriumph with Christina Schmitz and Jérémy Matou. They used a tactile discrimination task where the subject received 2 stimulation at different frequencies, and they had to answer this question: is the frequency of F2 higher or lower than F1? They compared other ASD people versus neurotypic. And what they found is that people with ASD, they had more precise prior, which is to be related to lack of generalizability, like a bit an overfitting model that would be less equipped to generalize to new sensation. And also they compared different contexts where F1 frequencies were picked from. Context, for instance, F1 could be picked in a rapid frequency versus slower frequency. And they were, when there was a switch between the two contexts, they could measure a slower dynamic of prior deaths in ASD people. There's another study by Lawson et al. They use Q target association learning task. They use, I will go fast. here, but what they found is they used a different type of models. It's called the hierarchical Gaussian filter. It's a 3-level model where the highest level enables assessing the volatility of the environment, how rapidly the environment jumps from one context to another. And what they could find with the behavior measurements is that ASD people tend to overestimate the volatility of the environment. And Lauriane, during her PhD, she used the same stimulation, and she could assess the underlying neurobiology with fMRI by confronting model prediction to bold signals. So now, oddball processing in autism. So Lauriane and Romain Bouet, they're actually, Lauriane did a large review of the literature of the MMN in autism, and they found it's really heterogeneous, the MMN in autism, contrary to other clinical condition, where, for instance, in schizophrenia, it's found systematically reduced in schizophrenia. In MMN, it's such a mess, and so Lauriane and Romain are trying to analyze this data quantitatively. And this heterogeneity pleads for a mechanistic approach for personalized diagnosis and treatment proposed by Helen Hacker a few years ago, which pleads for dynamic modeling. So we designed an experiment where basically we wanted to address this question of informative error versus uninformative one. So just to quickly understand what it is about, when we had this heat wave in 2003 in France, we say, oh, okay, climate, it's a random event, climate is random. So we didn't care, well, first speaking, these things could happen, no need to adapt. But now, since we have regularly every year heat waves, then now we think to consider that the rule has changed. And so we should revise our internal model and to adapt. So basically, this hypothesis we wanted to test, we have this oddball rule, and that in one condition, it keeps changing from one deviant probability to another. And we contrast it where another manipulation, where in this context, the rule does not change, but it's the precision of the rule that changes over time. And we hypothesize that in the two populations, there will be the same processing this condition, because both groups will keep tracking the change of rule over time. Whereas in this condition, we expect that at the beginning, they will process similarly, tracking the changes. But then after a while, the neurotypic participants, they would understand that the rule hasn't changed, and so they will slower their learning rate as compared to the ASD people that would keep tracking a change. So it's a collaboration with Laurent Motron in Montreal. We have this data. Kristen, who is here, performed the ERP analysis with Janne Reinders. And so we have a difference between groups regarding the mismatch negativity. It's reduced under ASD, but now we need to go to the modeling, and that will be from next year. So conclusion. Bayesian theories of brain function. So it's a framework accounting for perception and cognition, of great relevance in autism because, again, it provides new hypotheses. It requires dynamic modeling to be fully exploited. And yes, further efforts are needed because, as the review by Serrietz concludes, the field is currently young and relatively immature. Our suggestion that they provide in their review could provide following studies with rigor, potentially aiding in future understanding of autism. So thank you, and thank you to my collaborators.
Thank you, Françoise. Questions? No questions? So maybe I have one question. You introduced the precision weight and predictive errors, and you gave this example of living next to an hospital. You are not surprised by an ambulance going by. So is it something which referred to a state, or is it something which is really dynamic? In other words, is a precision weight something could evolve according to your understanding of rules, or is it a characteristic of a state, like, for instance, being ASD or not being ASD? You see what I mean?
Yeah. So the regarding the hospital, it's precision weighting is a dynamic state, and the idea is that it evolves slower than the prediction errors. So this is the way we can disentangle the two. And yeah, regarding autism, it's not. This is something we have to test, of course. This is the point of this kind of framework, is to assess how it evolves over time. The idea is that in ASD it does not evolve. We expect difference in the dynamics, and using hierarchical models, maybe we could see different, because there are precision weighting at different levels. So it's complicated.
Okay. No more questions. Okay, so I think we are almost in time for the break. And thank you, Françoise, again. Okay, so we start again for this second half of the session. And now we have Florent Gobert. He's a medical doctor. He's at the neurological hospital. He's a neurologist and intensivist in the intensive care unit. And he's working on EEG and coma and disorders of consciousness. So we will talk about metrics to assess consciousness dynamics.
So thank you for the invitation and the introduction. So indeed, I will present you the EEG metrics we developed to assess consciousness dynamics. So it was a neurophysiological challenge that we want to tackle during my PhD, to mathematize variability for neuroprognostication purposes. So after a short introduction of the medical context and the theoretical background, I will develop a little further the ad hoc methodology we developed here in this lab. And then I will present you just a few results from this methods, and then short conclusions. So about the medical context, the challenge of neuroprognostication is in fact a part of my common routine work. But if you are not aware of what we are doing in ICU, I will present just shortly what happens after brain lesions. So after brain injury, some patients can present an early recovery if they have small or non-strategic injuries, and they can evolve to brain death if they have very severe lesions and inefficient treatment. And in some cases, and most cases, in fact, they have large and strategic brain injuries which evolve to transient brain dysfunctions. And concerning the level of consciousness, the disorder of consciousness are one of the possible consequences of the brain lesions. And they are assessed clinically by a behavioral scale, which is called the coma recovery scale. And to represent consciousness modulations after coma, we use this very simplified view of consciousness with two dimensions, the level of wakefulness here and the content of consciousness. And we are not currently experiencing the dissociation between both dimensions because we are now in a conscious wake. Just a few hours before we were in a deep sleep, and both dimensions evolve concomitantly, except for the paradoxical sleep. When we are anesthetized, we lose consciousness in both dimensions, and patients in coma without anesthesia have the lowest value of both dimensions. And then they can evolve, for some of them, through a dissociation between the level of wakefulness, which can increase only, and they enter into the unresponsive wakefulness stage, which is the former vegetative state. And as soon as they present some sign of content of consciousness, which are in practically assessed by any sign of cortical function. They enter into the minimally conscious state. And then as soon as they are able to communicate, we call them the exit MCS state. So more theoretically, the definition of consciousness are usually based on what happened in the brain when we become aware of, for example, one picture, either depending on the experience, the global experience that we have in front of this picture, of the internal cognitive experience, for example, seeing a tree or having the sadness in front of a tree in autumn. This is a phenomenal consciousness. Then you can have another aspect of metacognition about this being aware of this awareness. This is the access consciousness, but this is the work for example, the Communicate Consortium about the correlates of these high-level mechanisms with the theoretical rivalry between the global neuronal workspace or the information integration theory. But for this study, we focused on the infraconscious prerequisite for these high-level mechanisms. And we assumed this paradoxical pivoting by not looking on the just intercortical connectivity, depending on this theory, or the thalamocortical connectivity, depending on the mesocircuit theory of consciousness, but we look at the prerequisite of the brain function, depending on for example, the brainstem or the encephalic function concerning the circadian rhythms and the homeostatic sleep pressure. So, in other words, we propose to segregate not only by awareness the two groups of patients, but also by the wakefulness features of these groups. So more theoretically concerning the fluctuations of wakefulness, after the transient brain dysfunction, some patients can evolve through a final brain dysfunction, and it depends notably on the neuroplasticity of the brain after brain injury, or on the balance with neuroinflammation and neurodegeneration with their own positive or negative dynamics. And in fact, the determinants of fluctuation, which are the circadian rhythms and sleep, are both biomarker of severity, we will see why, and adjunctive factors for recovery. In fact, there is quite a strict collocation in the brain of the determinants of the modulation of the cortical, subcortical loops by the pro-arousal system, depending on the brainstem, notably the reticular formations, which is in turn modulated by the pro-sleep system, notably according to the function of the anterior hypothalamus. And it's in the same areas that we have the circadian system. So, of course, if you have in this place severe brain lesions, you can have both the coma and the dysfunction of what could impair or increase neuroplasticity, which are the promoter of sleep and the promoter of circadian systems. So our scientific question was to assess in real life the fluctuations of this patient in ICU by shifting the temporal scale and the spatial perspective of doc analysis by using long-term assessment of at least 24 hours to objectify long-term fluctuations, to assess the natural history of coma by, when possible, longitudinal analysis of the same patients in a non-cortically centered approach using multimodal assessment of body rhythms, using hormones assessments and behavioral changes of eye opening. We have a prognostic hypothesis that the outcome would be more probably favorable if fluctuation were present early due to the pleiotropic favorable effect of their determinants, which are sleep and circadian rhythms. So we'll focus now more about the methods which were developed here in this laboratory, thanks to several engineers, Marc Thévenet, Samuel Garcia, and Alexandra Corneli. So we had recruitment thanks to the acquisition which was developed to co-register all these metrics. We had usually single recordings for each patient, except for 2 patients evolving from the acute to the chronic disorder of consciousness. We had 18 acute patients from 2 ICU in Lyon with a fairly balanced outcome. And the favorable outcome was defined by the ability of the patient to be both awake and aware with communications during 2 years of follow-up. So we use this micromet system to have both, using the polysomnography, the brain information from the EEG, the body information from the polygraphy. We had also a camera in front of the eyes to hide the body information of the eye-opening behavior. We also have, for patients, not for this healthy subject, the melatonin and cortisol hormones to have the consequence on the body of the circadian rhythms. And the environment, we cannot see here, but we have a lux meter and a sonometer to know what happened in the ICU room. So all of that was strictly synchronized using common triggers sent by a LabVIEW system and then extracted with a Python script to compare all of these assessments of behavior, hormones, and neurophysiology. And then we extract data with dedicated software. Here it's, for example, the Vulkan software allowing to extract manually the eye-opening behavior and the motricity of the patients by looking at each videos. We also extract with Python the lux meter results and sonometer results, and we extract the hormone assessment allowed with a collaboration with Véronique Ravreau from the hospital and Claude Gronfier to look at the fit of the melatonin and cortisol rhythms. Then, of course, I will have a focus on EEG results, and notably the pre-processing, because it was a real fight against the EEG artifacts from the ICU, which were mainly related to nursing, the changes of the muscular tonus related to brain lesions, the sweating, which was very awful, in fact, and the cough, and for example, also electrical environment. So we had 3 steps for artifact rejections. First, we choose a uniform high and low pass filter between 1 and 30 Hertz. Then we had an automatic exclusion of extreme values, and then very time-consuming manual exclusion of prolonged moments of artifacts, notably for the nursing periods. Using an artifact viewer developed explicitly for this study to allow the visualization of the raw EEG, then the presentation of the filtered EEG, and then the selection of the most artifactual moment with the Python script developed by Alexandra. Then we focus on the EEG extraction. We had the main issue because we had a first hypothesis that was not confirmed. It was that it was possible to extract and define hypnograms in coma. And in fact, it was based on the literature experience from chronic disorder of consciousness, but it was clearly a failure, despite the involvement of a sleep specialist who was Hélène Bastugi in this case. But as a consequences, we had to have a very careful visual analysis of FFT, which was the basis of the hypnograms we failed to perform. And then we we had the intuition that there is an association not with the sleep patterns, but with the fluctuations of FFT and the outcome. For example, we had this very seminal patients, the pilot patients that we were not able to keep in the last analysis because, in fact, it was too easy. It was already almost aware because at the end of the recording, it was able to respond to simple command. And it was possible to build an hypnogram for him. But we tried to record later a more severe patient where we were still intubated and earlier in the coma development. And so we were not able to perform hypnograms, but we observed these patterns of fluctuation here of the low rhythms with disappearance of low frequency band and then reappearance. And these patients have usually a more favorable outcome than this kind of patient with very few fluctuations of low rhythm and only fluctuation of high rhythms probably related to the artifacts. So we tried to mathematize these fluctuations by 2 levels of analysis. If you go back to the anatomical functional perspective, when you use EEG, you look at the cortical reflection of rhythms which are related to cortical subcortical loops. So we had first an analysis at the seconds levels, and then another analysis of the modulation of these rhythms, but what happens in deep brain structure, for example, diencephalon and mesencephalon and reticular formations. So we developed short-term brain signal analysis and then long-term fluctuation of these short-term brain signals. For the first one, we first pre-select some relevant metrics with Python development with the PyEEG library. And then we decide to choose some complexity assessment according to some mathematical features of complexity from information theories, for example, based on temporal organization, so one entropy matrix. We also look at the fractal geometry of the EEG using the DFA, the detrended fluctuation analysis, and the chaos system in dynamic, the chaos theory using determinisms. Just have a focus on this result. For example, the fractal dimension is the way you use the geometrical precision of your size of window to define more or less precisely the complexity of your EEG. If you use, for example, the relationship between the size of windows and the residual error, you can see that for a more complex signal, you need to have shorter size of window of analysis to have the same residual errors than for a less complex signal, which allow to have a longer size of window of analysis. So the higher the DFA, here the DFA is at 1.5 for a more complex signal, the shorter you need to have a size of windows of analysis. We now then developed the determinism, which is a way of assessing the probability of occurrence of recurrent patterns in recurrent plots. The determinism is defined by the existence of oblique line, which means that at different times of the EEG, you will have very similar patterns reoccurring. Then we can observe at the observational part of the study with dedicated notebooks each of the feature we developed, and then we select the most appropriate electrodes. Usually it was a medial line because we had less artifacts, and then we finally provide final selection to avoid redundancy. So we had the power assessment of EEG, very simple way of assessing EEG. Then a spectral analysis based on absolute and relative power, and also in collaboration with Christian Berthomier from the PhysIP company, the dominant frequency in the alpha band and an autoregressive model to have a global view on the spectral analysis in magnitude and frequency, and the spatial variability, which was computed by analogy to the global field power matrix as a standard deviation of the relative power ratio throughout the bipolar montage in the 4 bands. And this complexity matrix I already explained. Then we had to develop long-term fluctuation because when we had to summarize in some way what happens in 24 hours. So this is a comparison of 2 patients which have strictly the same behavior. They have absolutely no moment of eye opening in the 24 hours and very dissimilar EEG on the spectral analysis, with here just 1 period of change of EEG with low delta and higher frequency bands, and nothing happened in the other period, and this kind of pattern with very fluctuating patterns. When we try to look at the mean value of spectrum, it was not so much informative, so we tried first to look at the standard deviations, to look at the amplitude of fluctuations. It was a bit more informative, notably for the alpha and theta rhythms, and so we tried to develop as much as possible of this matrix. So first the mean of each value, which can be presented here, and also the amplitude of variability, which was the standard deviation and the ratio with the mean, the coefficient of variations. But also we tried to develop complexity at the complexity analysis, not for the EEG, but for the long-term EEG. So we had 2 levels of complexity analysis. For example, here you have an entropy which is low, because if you have the first part of the signal, you can predict what happened next. Contrary to this example, if you have just this part, you cannot predict what happened next. So the entropy is high in this case, either with a high or a low amplitude, but we interpret that also in the opposite way, because the lower the entropy, the higher the predictability, and the higher the entropy, the lower the predictability. We also analyze the rhythmicity of the signal by the probability of fitting with ultradian or ultradian pattern. The circadian is 1 fluctuation in 24 hours, and the ultradian pattern is several fluctuations during 24 hours. So now some few results to know what it can provide. First, what would be a good rhythm for recovering brain? Its results is still in the final review not yet published. So if we summarize what's the first step of analysis, we had 21 short-term EEG features, which were developed in Python. Then we analyzed the long-term results with 8 long-term variables per feature. So we had a combination of more than 150 neurophysiological parameters. And to select among them, which were the most useful, we use a supervised data-driven analysis by comparing in a regressive model which of these parameters were the most useful to predict clinical factors or paraclinical results. I will just present you 2 of them. First, what does it mean to have a disorder of consciousness concerning fluctuations of EEG compared to healthy subject? Here you have in red the patient with disorder of consciousness, and they have a lower matrix of period of fluctuations for EEG complexity compared to healthy subject. In fact, healthy subjects have a fluctuation of 24 hours, and almost all disorder of consciousness patients had a lower period of fluctuations. They had never a circadian pattern. The amplitude of fluctuations for several EEG spatial variability metrics was also lower. And then when we look into the group of patients with disorder of consciousness, the outcome was related to a more physiological patterns. Patients with a better outcome are in violet, and so we can observe that they have a higher amplitude of fluctuations in alpha power or in alpha spatial variability, and they have, it's quite tricky here, a lower complexity results for the long-term EEG assessment. So it means that there are a higher predictability of their EEG rhythms at the long term. So despite the loss of circadian rhythmicity, a good EEG rhythms is a broad and predictable fluctuation within high frequency bands, spatial variability, and complexity. When we summarize all of these factors into a single view, we observe that for the long-term variables, the mean value were never useful. We also have the information into the fluctuation magnitude, complexity, and rhythmicity, and most importantly, for the short-term EEG analysis concerning the spectral results and the spatial variability, and less extensively for the signal complexity. Short results about the correlations between each dimension. So what does it mean when patients open their eyes in the recovery process? The method was a little more simple because we just use the EEG features and compare them to the time series of other assessments. We have 4 levels of comparison. I will just present you 2-dimensional comparisons with the behavioral information recorded from the eye-opening periods. So what does it mean when patients open their eyes, in fact, in the recovery process after a coma? Here you have the population of patients who will finally recover. And when we observe that when their eyes were more open here, they had a significant decrease of the dominant frequency of their spectral analysis. So something happened at last for this patient, contrary to patients who will not recover. And they have an eye opening period with statistically less changes of EEG. So when there is an eye opening moment which is clinically relevant, it's when there is a concomitant change of brain activity. So it could be regarded as a new sign of cortical function associated with eye opening. So as a conclusion, this new perspective on EEG and the new methods we developed to assess them probably help us to validate the pivotal point we proposed in the introduction in the acute phase of brain injury by not tracking early awareness, but the condition of future possibility of awareness. And the long-term EEG methods help us to go as close as possible to the central regulator of the wakefulness modulations. And perhaps the shape of the good fluctuation, which are broad and predictable, have a sleep-like patterns. And this shape of fluctuation, contrary to this shape of low amplitude and complex fluctuation, remind us of the flip-flop model of sleep. The system is, when something is able to fluctuate in the brain, the system is either in one state or in the other one with the sharp changes. And what we can say is that when it's functional enough, it's remain abnormal because it's not a circadian fluctuation. They are not awake for 12 hours and then asleep for the next 12 hours, but it switches sharply and with rapid on and off switch as a quasi metronome of wakefulness. So thank you for your attention. I want obviously to thank all the people involved in this research, notably the engineer team like Alexandra Corneli, Jonas Aberno, who was a student, Marc Thévenet, and Samuel Garcia. Thank you.
Questions?
Okay, thank you. It's really a great talk, really interesting. I wanted to ask a question which is maybe not, goes a bit to the side, but very related. So if you know which features like are important for this close to wakefulness state or recovery state, do you know, is it possible to stimulate somehow in this moment some particular brain regions using some non-invasive stimulation to kind of...
You mean to use information of what happens during 24 hours to be at the patient bedside to stimulate him at this moment?
Something like that, yes. Using something
we would like, in fact, it's what we would like to do, but it's very difficult because we need to record the patient several days and to look at if there is a pattern of the moment in the day when it's the highest state, but it can be very irregular. So you can have a rhythms, but it's not at the highest level at 2 o'clock. Or you can have an alarm when he's able to be at the highest level, and so you come and make stimulations. It's theoretically interesting, but very difficult in practice. But obviously, it's what we would like to do. But the point is that it's so much rapidly shifting, so you can start your acquisition and then it changes in this moment. So the other point is to remain and to perform several stimulations in the day, and then to look if there is better moment and what is the EEG correlate in this moment when you observe the highest level of induced response.
Other questions?
Thank you a lot, talk. I don't know if I missed one point or if you explain it, but do you exclude that the changes that you see in all these features and the relation to the patient's outcome are related to awareness too, and not just to the circadian rhythm? Or
to awareness assessed by no. In fact, we are relating. Okay, we have not any other clues of awareness during the process. Mm-hmm. In fact, we are prognosticating, so we are looking at which are the brain capability of the brain who will evolve toward future awareness. Mm-hmm. So this is the dichotomy we provide for the perhaps just one response. We have not this. I didn't present that, but one of the features, one of these. We have here several factors. Okay, so I present just two of them. So we have dog versus healthy, and present functional the outcome. So this is an answer. We also look at what is the relationships between the fact of having a circadian rhythms according to hormones. So it's the factor hormonal circadian rhythms. And we also have the factor having a behavioral circadian rhythms, but it's only based on eye opening. So it's kind of objective assessment of being a responsive wakefulness syndrome. And I have something perhaps closer to what you say, it's cortical functions, but it's just patients with a single CRSR, which have been regarded as MCS. It doesn't mean that we look at the cortical function every 2 hours. So it's the next study. So we don't have this kind of granularity of the assessment. We just know that some patients were MCS and other patients were VS, and some patients had circadian rhythmicity of eye opening. So they are the truly vegetative state as defined by the consensus, which is never used, in fact, of vegetative states is not just eye opening once, it's just eye opening and then eye closing with a kind of rhythmicity. So we have this information, but just, for example, for the cortical function, just once in the recording.
Other questions? Maybe just one question. Since you have all the eye openings markers, did you look at the spectral EEG changes related to eye opening? And is it something
there
that,
is one reason for which I could analyze this study for still many decades, is that we have performed a smoothing at 10 minutes. So we don't have the granularity again. So we could, because it's very strictly synchronized, but we need to go back to another granularity and then to look at the reactivity of the process. So it was what I expect, but it's very difficult to perform all that in the same time. But we could look at that. But we need to go back to raw data it's not possible at this level of analysis.
Thank you. No more questions? So thank you again.
Thank you.
And the next speaker is Marcia Di Lucia. She's a researcher at the Department of Clinical Neuroscience in Lausanne, at the University of Lausanne and CHUV, Centre Hospitalier Universitaire Vaudois. And she's also working in the field of disorders of consciousness, looking for electrophysiological markers of this state.
Thanks a lot for the invitation. I will show you some of our studies where we look at multivariate decoding of EEG responses to sensory stimuli, mostly for predicting the chances of awakening of comatose patients. So I will show you some of our first studies where we look at the neural correlates of auditory irregularities of comatose patients at bedside. Then I will give you just a little glimpse on more recent work that we did on resting state EEG. And as you will see across all these results, I will show you that these EEG-based prognostic markers are extremely sensitive to the passage of time, especially during these first days after coma onset. Like this. So the data are collected in Switzerland across multiple hospital sites, but mostly in Lausanne and in Bern. And the cohort that we work on is mostly comatose patients after cardiac arrest. This is one of the main causes of admission of intensive care units in Western European country. It's a condition that has a survival rate that is still quite low. And in countries like Switzerland, in which the withdrawal light-sustained therapy is performed, the chances also of recovering with preserved neurological and cognitive functions, it's about 50%. So in this context, it's very interesting to develop prognostic markers that can be very accurate early on after cardiac arrest for two main reasons. One, the main reason is that we want to prevent the withdrawal of light-sustained therapy for patients who will have chance to recovery, but also because it's a very sensitive window where one can also introduce more personalized intervention and improve the chance of recovering. And the state of the art is that the clinical assessment are mostly tailored to predict patients that will not recover. And this is based on many assessments repeated over 3 days after the coma onset, after which, according to the current guidelines, the withdrawal light-sustained therapy can be performed if patients have low chances of recovery. On the first day, these patients are typically treated with what is called targeted temperature management. So the body temperature is lowered to a target temperature between 33 and 35 degrees. And they also receive a cocktail of sedative agents, after which the body temperature is brought back to normal. And this is also a reason why during the first hours of the coma onset, many of the tests that have been performed for trying to develop prognostic markers, this period has been neglected or has been kept aside because all these treatments are considered as confound. So when we started more than 10 years ago now, we decided to investigate better what happened looking at the sensory processing in these patients. Now, do they relate to the long-term outcome? Looking not only at variable latencies after coma onset, but also during these very first hours, even if the patients are treated with this temperature management and with sedation. And the reason is that during these first hours, we have a very privileged condition of investigation, despite these confounds. The privileged condition is that the brain of this patient is not lesioned yet in most of the cases. We are looking at quite preserved brain tissues, despite the patients are in a very deep unconscious states. So we can treat them as a quite homogeneous cohort, which is not the case on other etiologies, not the case when we look at them at different latencies. So this is our protocol. We record at bedside using 19 electrodes EG, the neural response to auditory stimuli, the same patients during the first and second day. And then we collect their outcome at 3 months based on the cerebral performance category. And we dichotomize this outcome based on this CPC as favorable if the patients survive, and or unfavorable if the patients die or if remain in a disorder of consciousness. And so, as you know, using a classic type of mismatch negativity paradigm, so we deliver sounds regularly paced and then interrupted by sound that can differ with respect to the pitch or the duration. In a normal healthy participant, this is what you would record. So it's a classic N100 response to sounds, and then a differential evoked activity starting at about 150 milliseconds post-stimulus onset in an healthy brain. In a comatose patients, and typically during hypothermia, this is what one could observe. So we see that these classic stereotypical auditory components are not there, and that the differential response between standard and deviant sound can occur much later than in healthy condition. This is just one example, but in fact, the pattern of response that we observe in comatose patients is extremely heterogeneous, and the latencies at which this differential evoked activity can occur varies a lot. So this is a typical context in which adopting this multivariate type of analysis is particularly suitable because it can really take advantage of the specific patterns of differential activity. And here I give you just a brief summary of one of the techniques that we developed for analyzing this data. Essentially, we consider each single recording separately, and we aim at modeling the voltage topographies in response to standard and deviant sound. We use a mixture of Gaussian models that provide, after optimization using cross-validation procedure, provides a way to label each voltage measurement for each time frame in each trial as a series of posterior probabilities. Each posterior probabilities tell us what is the chance that each single measurement is well represented by one Gaussian of the mixture. Another way to represent it is to label each of these measurements using the color that label each single Gaussian in the mixture. And as you can see also in the average posterior probabilities across trials, typically these Gaussians tend to cluster over time, not only over space, which also gives an indication of the fact that this mixture represent or resemble the classic sequence of components in response to a sensory stimuli. And this doesn't provide only a representation, but also can be used to build a discriminative functions. And this is what we use for this type of application. Essentially, we compare the posterior probabilities for each of the Gaussians, and we derive the time frames over which we have the most information for labeling single trials in response to sound type. At the end of all this, essentially, we have a decoding accuracy, which tell us how well at the brain level the single trial that we record give us information of the different pattern in response to sound type. And what you see in the following slides is the area under the receiver operating curve of this decoding performance, which is a proxy of this auditory processing in response to these regularities. So I show you again these exemplar comatose patients where we run this analysis, and you can see that at the end, not only we have the decoding accuracy, but also an information about the time frames that the algorithm found as most informative for decoding standard and deviant responses, and also the topographies that are associated to that. And this can be used also to try to interpret at what stage of this auditory processing this difference between standard and deviant sound has been processed. So in one of our first studies, we collected data in 110 patients after cardiac arrest during the first and second day. We could include in the final analysis 94 patients within which 56 had favorable outcome. I don't show you all the results of this study, but essentially what we found is that irrespective of their long-term outcome, many of these patients exhibited accurate decoding in response to standard and deviant sound. Here I show you a qualitative summary of these results by averaging this area under the receiver operating curve of the multivariate decoding during the first and second day and grouping patients with respect to their outcome. So when trying to relate these values at the single patient level to their long-term outcome, we found that there was no clear cut between having preserved auditory processing and chances of awakening. Instead, we developed a new hypothesis based on these results, which assume that is not the preservation of the sensory processing that inform about the outcome, but is the temporal dynamic of this preserved response between the first and the second day. And so we computed for each single patient the difference in the decoding performance between the second and the first day. This is what you see here. Each point is one single patient. And you can also see that the vast majority of patients that improve from the first to the second day are those who survive. This leads to a positive predictive value of 82%, based on the fact that we observe an improvement in 33 patients, within which 27 survived. We can do better than this by identifying some patterns in the EEG that were common to patients that were not correctly predicted. We look at the EEG, the continuous EEG of these patients, and we found out that by disregarding those who exhibited epilepsy for discharge, which are very typical in patients with unfavorable outcome in this very early phase, we could improve significantly the positive predictive value and identifying early on survivors during the first 2 days of coma. And so this type of test can be used to complement the existing assessment in the clinical routine that is mostly informative about the negative outcome. In the next step, we also try to refine how the patients will actually survive, characterizing better their neurological cognitive states. And for this, we zoomed only into the survivors, those who could both be tested early on using this auditory protocol, but also those who could be assessed based on a large battery or neuropsychological test at awakening. We group the survivors as those who could be predicted based on the auditory protocol, so those who improved from the first and second day versus the others. And if we compare these two groups based on the score of this neuropsychological test, you see in this quite dense table that this group of patients differ in many of these tests. So those who could be predicted are also those who wake up and with better cognitive functions. Another way to look at it is to correlate the extent of the auditory improvement between the first and second day and the scoring of these tests. So the better was the improvement, the higher were their performance in phonemic and semantic verbal fluency. And also there was a negative correlation with the coma duration and with the hospital stay duration. So overall, I show you that by assessing this early on sensory processing, one can get information about survivors at 3 months and inform also on the functional cognitive outcome. It's a test that encourages the life-sustained therapy, so prevent withdrawal life-sustained support, and also can be applied without pre-inclusion criteria, so can be made automatic. And now I switch to more recent type of investigation that we are performing using higher density EEG in the same type of patients. This time I show you the results when applying deep learning in resting state EEG. Again, we record patients on the first and second day after coma onset. Results that are summarized here are basically optimal, so much better than we could do up to now based on assessing sensory processing. The AUC here refer to the area under the receiver operating curve or the prediction of the outcome, not anymore about the multivariate decoding of the auditory response. And you see that both in the train, validation, and in the separate independent test set, we basically have almost optimal performance. Here is the visualization of one of the folds in the cross-validation. You see that we have only 4 incorrectly predicted patients. And this is based, again, on the first day of the recording using this quite large data set. And in this case, we can also predict not only the survivors, but also those that do not recover. For those for which we could run a second recording on the second day, we see that the results are also very promising with an accuracy of 72%. Nevertheless, there is a significant drop in the prediction performance between the first and the second day. And this is not simply due to a smaller cohort of patients, because results also hold if we keep exactly the same patients on the first and the second day for reasons that we're still trying to investigate. And another nice feature of this type of analysis is that we can also use the this GradCam representation to retrieve in the raw EEG signals what were the temporal features that were used by the deep learning analysis to weight and give more confidence to a decision towards favorable or unfavorable outcome. So here, for example, in the second panel, you see that the weight for unfavorable outcome in the second bar is concentrated along the epileptic for discharge that are known to be what are called malignant pattern and normally indicative of a bad prognosis. For favorable outcome patient, this weight is more widespread because it resembles more like a normal EEG. And one could go and see across all the different recordings what can be informative. And finally, we also look at more intuitive type of features, global features of the EEG, such as power spectra, whether the confidence of the deep learning analysis could relate to that. And basically what we extracted is the power spectra between 5 and 15 hertz that we separately assessed to be quite informative about the difference in favorable and unfavorable outcome patients. And you see that only on the first day, we found a positive correlation, significant correlation between the decision of the deep learning and the power spectra, suggesting that when the prediction is optimal, this also use some of the features that we can intuitively understand also in the signal, which is not the case on the second day. So as a summary, I show you that by simply looking at resting state EEG, we already obtain a lot of information for the prediction of this long-term outcome. I didn't show you that we also have evidence that complements existing clinical tests and can support the decision already on the first day of coma. And this opens also lots of questions. So we're working now on other etiologies, see how does it generalize to different type of coma. And of course, understand better what is so informative about these first hours of the coma onset and why this information then is lost over time. And with this, I thank all the people who did the work and collaborate with me. Thanks.
Questions?
Thank you for the talk. First, I think it's very difficult to work on anoxic patient because we have already a lot of biomarkers of evolution, and patients with unfavorable outcome have probably death because of neuroprognostication. And so what is the independency between your markers and the markers which have been used for the to decide withdrawal of life-sustaining?
Yes.
Because when we look at the raw EEG, okay, it's a malignant patterns. Probably it's difficult to have a local global informations and auditory regularity with this pattern, but we already have the informations. So do you have a short, less important cohort of patients with for which it's difficult to know if they will evolve, and because for the patient with a clear-cut prognosis, we don't have much information to give.
Yes, I agree. So this is a very important question, and as you can imagine, there is a lot of discussion going on this. So we run two different type of tests to understand this. So essentially, in the clinical practice that we see in Switzerland, and this might be very different in different countries, because this is very much affected by how these markers are used in the clinical routine. But in Switzerland, we have about 30% of patients where there is a quite high chance that the patients will not survive. Now, in this case, life-sustaining therapy is withdrawn. So we will never know, right, with the current practice, whether this is the case or not. But let's say that these are the trivial case. Then there are some patients who wake up very quickly, actually. In 2 days, they are up. Then there is a quite large percentage of patients that are in the so-called gray zone. And the gray zone is typically the case where all these markers do not agree. Because it's true there are many markers, but all these markers are suboptimal. And these are suboptimal, especially with these patients for which we don't know exactly. You know better than me, the neurospecific anulases, somatosensory evoked response, this malignant pattern, the brainstem reflexes, etc. So what we did in the first studies with the auditory processing, we tried to zoom in those for which these markers do not agree. And these were either patients that had only one bad prognosticator, or those who had no bad prognosticator but did not wake up, and no withdrawal assisted therapy was performed, which left us with about 30 patients out of 94. And in this, we had 13 for which the progression of auditory performance would have been informative about survival. So this gives you an idea of the type of information that we can get as a complementary. For the rest in state EEG, the story is completely different. So what we tried was to dichotomize the patient, not with respect to their long-term outcome, but with respect to whether they presented or not a good score as a result of the prognostic markers that already exist. So, for example, presence or absence of brainstem reflexes, presence or absence of motor response. You know, there are many of these things. And the thing is, the deep learning does overlap with many of these markers. But, for example, for the brainstem reflexes, the information that it gives is almost orthogonal. There is no relation between the brainstem reflexes and what we measure here, which might be obvious. I'm not sure, but brainstem reflexes are used to take the decision. So the relation is not so obvious. Now, to derive the gray zone and really the complementarity, we've been discussing this, I think, for the last 2 months. There is not 2 doctors that agree on how we define the gray zone. And I think this is already tells quite a lot of the story. And now these decisions are taken is really very much individual and subjective decision that is taken. This is my view, but I might be wrong.
Thank you for the really great talk. I wanted to ask a more technical question specifically about your deep learning study. So the accuracy looks so great.
Yes.
A bit. So I just wanted to understand how exactly did it happen? Like how precisely did you do the cross-validation there? And if you did it across patients, did you do some effort to align somehow the recordings of different patients together so that the algorithm works?
So I should first say that I didn't run the analysis myself. I know quite a lot of details about it, but I'm not super expert of this deep learning yet. I'm trying to learn. So essentially what we do, we have these recordings in 8 and 20 minutes. Then we epoch the recording. We have 5 seconds epoch, and the deep learning works on the 5 seconds. So at the end, we have a confidence that is assigned to the 5 seconds. And for the patient, we take the average of the confidence that is obtained on the 5 seconds epoch. Each patient is treated in a way that in the cross-validation, if the data of the patient is in the training, will not be seen in the validation and the other way around, and similar for the test. So the data of single patient is not splitted in the different split of the data. So each new patient is really completely independent. And it is quite miraculous, this type of results, but I have to say that this has been shown already by other groups, so we're not the only one. Our novelty is really that we look on the first day, which is this neglected time where people don't look at because of this sedation and this type of treatment that the patients receive. It's but the EEG, resting state EEG, is very, very informative in this case. I don't know if I answered your questions. Maybe more about the architecture of the deep learning.
But like, basically, my question is: can you apply this to new patients if you train a network on?
Yeah, the test set is completely new, and the results that you see here on the test is based on completely new test.
Awesome, thanks.
That's a really interesting talk. I have a very naive question. So if I understood correctly, all of those patients have this medical procedure going at lowering temperature and sort of pharmacological cocktail. Maybe what you see is the pharmacological response to this, and this has some pattern in EEG, and then it sort of works down after that second day.
It's not a naive question. It's a very pertinent question. We would love to investigate this. To do that, we would need the same cohort without the sedation, without the which we don't have. So what we did in one of the simpler study that I don't show you here, we look at just the power spectra on the first and the second day. I think it's the only domain where you can still publish something using just the power spectra. So I invite you to profit from this because it's just unbelievable. Nobody looked at the power spectra during the first day of coma for this before. But so we just look at whether the sedation could explain the difference in power spectra on the first and the second day. We ran a multilinear regression model, there is no trivial explanation. But because they receive a cocktail of these sedative agents, they have fentanyl, midazolam, and propofol, it could, and in this model we regress out them separately, could still be that there is an interaction effect that is difficult to model because it's in the physiology of how the brain responds to it. But just to say that is not so obvious. And also we look at whether we can better classify those that are more sedated versus those that are less sedated, because sedation is based on the weight. So there is some variability, and also based on some agitation of the patients. And again, there is not such an obvious relation. So this gives an indication that it's not trivially related to that, but it might be, and might be also an indirect consequence of this. Yeah.
Okay, no more question. Thank you again, Marcia. So our last speaker is Perrine Séguin. She's an MD in physical medicine, and she's doing her PhD with Alessandro Farné, Jérémy Matou, and Emmanuel Mabi at the CRNL. And she will talk about disorders of consciousness and the usefulness of BCI in these conditions.
Good morning. Thanks, Olivier, for the introduction. So yes, my presentation will question the usefulness of BCI in patients with disorders of consciousness. So I start with a little definition of brain-computer interfaces. The subject tried to make a command with his brain, like imagining moving his hands. And we detect the signal, we classify it, and we can have many different kind of effectors. And importantly, to be a BCI, the device has to include a feedback to the user to allow the user to learn from his brain activity, if he's made an error or not. This device, most of the time with patients, they work with a calibration first, and after they can be used with a test phase. So for patients with disorders of consciousness, what could be the use of brain-computer interfaces? As you realized from the previous sessions, there are a lot of limits of the behavioral assessment. So this motivated the rise of active paradigm. You saw previously mostly passive paradigm, where brain activity is recorded, where we ask nothing to the patient. But there are also active paradigm where you ask something to the patient, moving his hand, counting some sounds, and you record it, and you can see if there is a modulation specifically due to this command. If there is this modulation, then the patient is considered as responsive to command covertly, so it's a particular case of cognitive motor dissociation. This is the name of this peculiar category of patients. It was discovered in 2006, first with fMRI, and it is now known that at the acute phase, cognitive motor dissociation is a factor of better prognosis. And it appears that even in chronic patients, 10 to 20% of patients with disorders of consciousness are in the state of able to respond covertly to commands. So the next step would be for this patient, obviously, it's a terrible condition, right? So we would like to restore communication, and BCI could be useful for that. It was first attempted with fMRI, and it appears that only 5 out of 54 patients could follow the command covertly, and only 1 patient could answer 6 questions. So already there is a gap from the response to command to the communication. So what is the difference between paradigm for response to command, active paradigm? I will talk about mainly paradigm with EEG now, because it's much more practical for this patient. So for the active paradigm, we only look at the electrophysiological response to command. For brain-computer interfaces, you know, there is an additional setup with an automated classification, a real-time loop, and you can have most of the time more commands because it's more practical for communication. So there are many similarities between active paradigm and brain-computer interfaces. We use sensory motor rhythm in both cases. We use evoked potential, P300. We modulate them by attention, both in BCI and active paradigm. And there is an increased automation of active paradigm analysis thanks to the expansion of machine learning, which make them closer to brain-computer interfaces. And obviously, the most practical hardware in both cases is EEG. And we finally target the same subgroup of end user, namely the patient in cognitive motor dissociation. But there are some additional features for BCI. It's often a prerequisite to have a more number of commands, and there is also, if you want to communicate, the patient has to be able to spontaneously engage in a communication, whereas for active paradigm, he just have to answer and follow the command, which is cognitively different. So there was a recent review that give a nice overview of all the BCI trials with patients with disorders of consciousness. And it appears that most of these studies try first to use it at an assessment tool. You see, it's really a great majority that does that, whereas it provides very few added value compared to active paradigm. It was less validated. So probably this is done this way because they address the vast majority of patients with disorders of consciousness, so they do not know who is in a cognitive motor dissociation or not. So they have to detect them, and after to start the communication mode. But the problem is very often it's difficult to calibrate these brain-computer interfaces, and especially to have a robust calibration to start a robust test. It's really difficult, and most of the time this step is not successful. Also, this review emphasized that there are major methodological limitations in these studies, very small number of patients, very heterogeneous qualities of clinical evaluation, and the statistical assessment are not always very robust. And this review excludes studies with patients who emerge from minimal consciousness or locked-in syndrome. So we do not know from this review if it's efficient for them or not, which would be important to know. So is it easier to control a brain-computer interface when you're conscious? It is not so obvious. So there is a review that addressed 73 studies, and you can see most of the time it's more patients with motor neuron degenerative disease, so a little bit different from the one we address. But there are some patients with stroke. Here, all the patients are conscious, and again, it's difficult to interpret because there are many methodological limitations. Most studies test feasibility. There is no guarantee that negative results are published because these are mainly proof of concept studies. There are very small numbers of patients, and again, the clinical profile is badly described. And the results are very heterogeneous, and there are a lot of needs for BCI adaptation in this conscious population. So you can imagine this is really difficult when you start to address patients with disorder of consciousness that are non-responsive. It's difficult to adapt the BCI for them. Also, what we realize is that very often extreme motor impairment, including oculomotor impairment, is associated with lack of BCI control. And so this is true mainly for this patient, but we know in this group from our experience and from some data in the literature that some patients fail to control brain-computer interfaces too. And we suspect that this group of patients could have more oculomotor impairment than the other and more global motor impairment. And that this could lead to attentional impairment. And as we consider that brain-computer interfaces are controlled mainly by attentional modulation, whatever the features. We suspect that this kind of motor impairment could also be correlated with poor BCI control. This is also confirmed by a lot of data in the literature, mainly fundamental literature on attention and oculomotor control. And this is true, whatever for overt attention when you can move your eyes, and also covert attention when you do not move your eyes. So we designed a study based on this hypothesis, where we compare the oculomotricity ability with an eye tracking and the attentional abilities of the patient with electroencephalogram. We also monitor a lot of factors that can influence the results. And we included a control group with healthy subjects, some patients with tetraplegia who can talk and and move their eyes. Some patients with a locked-in syndrome, so these patients are strongly paralyzed. They cannot move, cannot talk, and they can move a little their eyes, enough to communicate, but not very precisely. And we had one patient close to disorders of consciousness, actually, who was not very responsive behaviorally. So you see there is different etiologies. What interests us is mainly the functional state of this patient, paralyzed or not, oculomotricity or not. We assessed 3 active paradigm with sustained auditory attention that was developed in the Lyon Neuroscience Center, where subject has to first imagine moving in his house when listening some sounds, and after he has to specifically focus on the sounds and count some different sounds. We also assessed another paradigm developed here, brain-computer interfaces, that is auditory, where the subject has to focus on one side, on the yes, and this allows him to select the yes. And finally, we use a classical paradigm of motor intention. The subject tries to imagine he moves his hand, he is at rest. In parallel, we assess the oculomotricity of the patient. Thanks to this kind of paradigm, the subject has to look at the cross and to fixate the target and to go back to the cross, etc. And after we use this kind of smooth pursuit paradigm, the subject has to have his gaze very precisely on the target, and this allows us to monitor the precision of gaze trajectory. Here, it's a more cognitively complex paradigm. The subject has to focus on the cross, has to fix the cross, sorry, and to focus on the lateral side and to count the different ellipse appearing only on one side. So it's a selective spatial attention, and it's close to what is done in BCI, what we do in BCI auditory. And we record the EEG also during this task. So these are the preliminary results. So here you have the results for EEG with one is when everyone could do the task in the group. So you see in yellow, it's a control group. Everyone is in the control group had a good control of the auditory brain-computer interfaces, a good selective spatial attention, whereas you can see for the locked-in syndrome person, it's already more difficult. And actually, most of the tasks are more difficult for locked-in syndrome person, even some tasks on covert visual attention. And for the results on the eye control, you can see that for the simple task, actually, it's like all the group perform almost the same. Whereas for the most cognitively demanding task, there is an important difference between the control group and the patient in their globality, whatever in tetraplegia and in Locked-in syndrome. So we have to analyze that. And you can see that the peculiar patient that is non-responsive, so close to disorders of consciousness, was unfortunately unable to control any of the EEG paradigm. And whereas he was able to follow a little bit the target we presented him. And he was, on the contrary, unable to focus on the cross when we asked him. He was like grasped by the target and not fixing the center. So some conclusion, BCI testing of patients in disorders of consciousness are faced with several layers of uncertainties. So some doubts concerning the sensory abilities, the cognitive abilities, fatigue, depression, motivation, same for all paradigm and behavioral assessment. There is an additional layer of problem added with brain-computer interfaces. We do not know, you have seen from our preliminary results, it's dubious that this brain-computer interfaces paradigm can truly overcome motor disability. There are more complexity in general, whatever the constraint and the cognitive requirements. So at the moment, there is no evidence that in first intention, BCI are useful because active paradigm are more robust, better standardized, much better validated, and more practical. You need less expertise to put it, and it can be analyzed after. There is still a theoretical advantage for communication, but there is a need for multidisciplinary expertise. There are very rare cases in the world of non-responding people that could use brain-computer interfaces longitudinally, but these people were mainly in motor neuron disease rather than in disorders of consciousness or even in classical Locked-in syndrome. And even in these very rare cases, communication remains limited to yes-no code and very fluctuating. Even one of the study, this one is an invasive one, and even in this case, it was fluctuating. So thanks a lot to the team that supported this work, and thanks for your attention.